So my favorite Halloween song is Flying Purple People Eater, but I digress. All right, this is Norse Teaches Science. Another biology edition will continue into our genetics information. And today, Mr. Norse, that's me, will present codominance and we'll solve codominance problems. So by the end of this video, I can explain how codominance can affect the results of a Punnett square. Yes, we're still doing Punnett squares. Genetics is full of them. You're going to do Punnett squares forever when you're in biology class. So codominance. What codominance means, if you just think of the word with the prefix co, if you don't know, like think of co-president or co-chair, it's when things are shared. So you're going to share dominance here. So in codominance, codominant alleles results in the expression of both traits. Both traits. So if you can look over here, this funny Punnett square, you have a duck and you have a beaver, and when they mate, which they can't because they don't have the right chromosomes or parts, um, results in it looks a like platypuses. I don't know what the plural is. All right, but as you can see, they're sharing the beak and the tail are codominant, so that's how that works. Um, you can also see like flowers that have both white and red on them, or fish that are striped with the two different color fish. But since both are dominant, they're both going to have their own letters. So when you get into codominance, this is where we won't have any lowercase letters. We'll have all capital letters, and you're going to have two different capital letters. So if we look at flowers, say we're this red and white flower over here, you're going to have two traits, red and white. So we're going to have a capital R and capital W. So capital R is red, capital W is white, and RW is going to be red and white. Or sometimes they say spotted or striped or something like that. They're going to say something about it. You're going to have to talk about both traits. And usually when you write them, you're going to write them in alphabetical order. So let's do an example. It says, let cross a red codominant flower with a white codominant flower. So the red flower would be capital R, and the white flower would be capital W, capital W. So we're just going to write them in our Punnett square over here. Then you're going to bring them down just like you normally do. R, W, R, W, R, W, R, W. So, everyone followed so far. If you didn't, rewind the video and then watch it again. So, genotype ratios, the first one I see is R, W. How many boxes have RW or both of the letters in it? All four. So that's 100%. And what is the phenotype ratio? Remember, you could have red, white, or you could have red and white. Do we have any genotypes in here for the red flower, big R? No, that's 0%. Do we have any WWs in here? No, that's 0%. The red and white would be the heterozygous, that's RW, and that's all four, so that's 100%. So this is the first practice. Now remember some of this stuff, we're going to use these same things in the next practice. This time we're going to cross two red and white flowers that are codominant. So what is the genotype for the flowers? That would be RW because they're red and white. So we're going to place RW at the top of the Punnett square, RW down the side. I want you to pause the video right now. I want you to try to solve the Punnett square and find the ratios. I'm going to count to 5, and then I'm going to begin. 5, 4, 3, 2, 1. All right, let me solve this. In this first box, we're going to bring down the R. We're bringing the R over. Next box, bring the W down, bring the R over. We get R, W. This next box, bring the R down, bring the W over, R, W. And this last one, W down, W over. All right. Genotype ratios. First one I see is big R, big R. How many boxes? 1. That's 25%. The next one I see is R, W. How many boxes? I see 2 boxes with RW. That's 50%. And the last one I see is W, W. How many boxes is that? There's 1 box. That's 25%. Do you guys see, if you've been watching these videos, I talk this out as I do it. That's something you should be doing, even if you don't do it out loud. In your brain, these are the questions you should be asking yourself as you solve Punnett squares. All right, what are the phenotype ratios? First thing, we need to identify what our phenotypes could be. You could be red, you could be white, or you could be red, white. When I usually make questions like this, I usually say it's like striped or spotted, so I would write striped or spotted so I don't have to keep writing all this out. So red, to be a red flower, you have to have 2 R's. How many boxes have 2 R's? 1. That's 25%. To be white, you have to have to be a white flower, you have to have 2 W's. And that's only 1 box, so that's 25%. And the last one, to be red and white, you have to be RW, and there's 2 boxes of that. That's 50%. So if I asked the question, what is the probability of these flowers producing a white flower, you would say 25% probability. And that is about codominance Punnett squares and how they work and how it affects everything. There's more Punnett squares that we're going to be covering in this next series. I have 2 more types we're going to cover. One's about blood types and one's about something called sex-linked traits. But I hope you learned something today. If you want to know more, go check out the Google on this topic. And like always, this is Miss Norris, and I'm signing out.